go. So as Denise mentioned today, I will be discussing some of my current research, and I want to first acknowledge the Institute and say a big thank you for having me here today, and a thank you to all of you joining for accommodating my English presentation today. I really appreciate it. Today I'll be discussing how we can use data science to tackle some of the most pressing questions in the ocean, everything from aquaculture to harmful algal blooms. And then I will end today talking about the project that is nearest and dearest to me, sea turtle migration in a changing ocean. So we have a lot to discuss today. Before we get into the research, I just want to cover a little bit of background about myself. As mentioned, I am a senior data scientist and currently affiliated with 3 different universities, Stanford University and the University of California. I spend a lot of my time primarily at Stanford University, but I actually live in New Zealand and spend the other portion of my time working for the Cawthron Institute, which is based on the South Island of New Zealand. I've been here for about 5 years. And the majority of my work, my main focus throughout my career has really been on species distribution for risk assessment and species management, with a particular focus on sea turtles. But since I moved to New Zealand, I started doing a little bit of work in the machine learning and artificial intelligence space, in particular, figuring out how we can use near real-time data sets from the ocean to monitor and provide ocean intelligence for management and decision making. Throughout all of my research, one of my favorite things to do is to provide data visualizations and effective science communication to a wide variety of audiences. That's the thing that I really enjoy the most in what I do. But it would be helpful for me to also acknowledge my little fellowship team that supports me in everything that I do, and that includes my husband Drew and my dog Betty. We spend a lot of time when we're not at the beach, we're hiking the trails in Nelson, New Zealand, and my 2 COVID adopted kitties, Olive and Tom, which I think this picture sums up their personalities pretty well. So Olive has a tendency to join Zoom meetings, so you may see her run by the screen, just as a warning. But let's actually get into the research and what we're going to talk about today. We're going to cover 3 very different research questions. First, we're going to start off with aquaculture in New Zealand, and then we're going to move into harmful algal blooms and seafood safety, especially under different global climate change scenarios. And then finally, we're going to end our talk today with sea turtle migration and dynamic corridors in the North Pacific Ocean. So thinking about New Zealand, it's a fairly small country. However, its exclusive economic zone, which is the portion of the ocean to which New Zealand has exclusive rights, is actually 15 times the size of its landmass. And given this vast amount of ocean, almost 4 million square kilometers, there is a strong incentive to move into the blue economy, which is effectively the economic use of ocean resources in New Zealand's waters. The primary industry for this is aquaculture. So in New Zealand, aquaculture is currently a $500 million a year industry. And there's a strong demand to move that number to almost 3 billion by the year 2035. So going from 500 million to 3 billion in 2035, which the year 2035 sounds like a far off futuristic date, but it's actually only about 11 years away from where we are currently. So given this, there's a strong focus to expand and to scale up finfish and shellfish in particular. But in order to meet these demands, the question is how can we incentivize and innovate in ways that allow farmers to catalyze their growth, to maintain the health of their farm, and to reduce the operational costs and environmental footprints, especially in a changing ocean. Now, as we know, managing for ocean resources is inherently difficult because the things that we're managing for sit beneath the surface of the ocean. So we can't go out and see everything that we need to manage. For aquaculture in New Zealand in particular, there's an added challenge in that many of these farms are 2 or 3 hours away by boat. And so it's significant time and fuel to be able to get out to an individual farm and monitor the health and its crop. Given that the ocean is dynamic and constantly changing, there's an additional challenge in how do we actually manage these farms in real time. And from interactions with the industry, we've identified that there's a strong need for basic data, basic information on things like water temperature or the ocean state. So is it windy? Is it are there big waves? Can the farmers actually access their farms when they need to? So that's where there's a strong desire to bring data into this industry. And in an effort to enhance the industry, the New Zealand government has actually awarded the Victoria University, which is based in Wellington, the nation's capital. It's awarded $13 million over the next 7 years to effectively deliver data-driven management solutions for building the blue economy. And so in a partnership between academic, governmental, and commercial industries, there is a goal to bring cutting-edge data science technologies to the aquaculture industry and to make ocean intelligence more accessible to everybody and effectively bringing the farm to the desktop. So really allowing farmers to see their farm in near real time without having to go out and consistently check on it or not be able to check on it based on ocean conditions. So the next slide, I'm going to show you just some of the topics that are included in this data science program. And the screen itself, there is a lot happening on this slide. So I just want to highlight with each group. I'm not sure if my pointer is working, if you can see it, but what I would like to just showcase is that there's a wide range of data science projects happening under this research program. Everything from developing deep learning models to score the skeletal health of salmon from X-ray images, to things like predicting breeding outcomes based on genomic variation for finfish. There's also a push to use deep learning and AI models to effectively generate harvest assessments to know when exactly farmers can harvest their crops at the right time. And I'll return to this one in a little bit. But the one that I've been mainly focused on is this use of transfer learning for object detection. So transfer learning in computer vision is the use of a pre-trained model. If you're familiar with convolutional neural networks, it's the use of a model such as that, where you can actually use a pre-trained model as a starting point for object detection to solve other similar or related tasks at hand. So the idea with using transfer learning is that you can take a large amount of training data, but not necessarily have to manually go through and label each exact image for the training data. It's pre-labeled for you. So I'll show you a little bit of what I mean by this as an example. So if we were to look at a mussel farm, a standard mussel farm in New Zealand, the mussels themselves, this is an underwater image on the left here, the mussels grow along these series of long lines that are suspended vertically throughout the water column. These long lines support thousands of meters of crop. So you can see that on the surface on this right-hand picture, these long lines essentially are held in the water column by these plastic buoys, these black sort of surface objects. And these buoys essentially form the backbone of these long lines of mussels that snake up and down in the water column for several of kilometers across the ocean surface. Now, the buoys themselves are integral to the mussel growth because they keep the mussels growing at an optimal temperature and depth in the water column. But the concern is that as the mussels grow and they get bigger, they start to pull down on these lines. And what happens is that the buoys begin to submerge because they start to take on water as the mussels get heavier and grow and sink deeper. The problem is that if the buoys take on too much water, they begin to submerge and they can actually snap off the line and go missing, or they can drag down the mussels with them. And in an instant, a farmer can lose tens of thousands of dollars in crop just by a buoy effectively sinking too quickly. So the process of going out onto the farm and assessing the conditions of the buoys is actually a really important part of maintaining the farm health. Oftentimes, farmers have to go out and regularly check if a buoy is sitting at the right depth below the surface. And if it's starting to sink, they need to add more buoys to help float the rest of the mussel line. So the loss of buoys is actually a really important factor in maintaining the economic sort of viability of the farm. So the question that we were brought by the farmers is, how can we help the farmers maintain the quality of their structures in their farms? So we've actually produced a few papers, some analyses that I'll give a quick overview on with this section that really identifies ways that we can help farmers remotely monitor their farm in between visits by using object detection for their buoys on the farms. So what we've been doing is we've set up several cameras around the farms that actually just video the buoys all day and every day. So we can get information on where the buoys are in each frame. And there's really sort of a stepwise process with this, is using computer vision to capture videos and to automate the process of detecting stationary buoys within each individual frame. But then the larger goal moving forward is to be able to track those buoys in the water column, especially if they begin to sink. And we can get an idea of how high up is the buoy sitting above the surface of the water. And if it is starting to sink with time, then the farmers know that they need to go out immediately and add additional buoys in the area or modify the structures. So one of the challenges that we faced in effectively doing computer vision or object detection on these buoys is that in some conditions, you'll see in figure A here, the sky and the water actually look fairly similar. And this can be really challenging when we're trying to basically label certain parts of an image to identify where the buoy is relative to the water or to the horizon. So the first step that we had actually was to segment out the image and figure out what is water and what is not water with this. And that was further complicated by cloudy days, or if you can see in images B and E, there's some solar glare on the surface of the ocean, which we have to account for. Or in image D, you can see that maybe there's the camera itself is obscured or has some raindrops on it. And I think probably a good analogy is to think about everything that goes into developing a self-driving car. And while that is a much more complicated endeavor, even thinking about how to tell the car when to look out for objects, when is there solar glare, when to stop. Those are the similar sorts of problems that we were facing, even just on a much simpler level with some of these images. So the first step was to remove the horizon or the water line here, because by doing that, we can effectively reduce the computational cost of looking at each individual pixel and identifying if it's where the buoy is and if it's water or not water. So that was step one. However, it was really difficult because of these variations by each different sort of ocean condition and sea state. So what we did is we ran a pre-trained model, and I will point you to this paper here if you'd like to learn about the modeling approach in further depth, this McLeay paper here. But what I want to show you is that we use several different types of models. We ground truthed it, and then we also used a pre-trained model with no additional sort of training after the model. We just plugged everything in and ran it versus refining that and fine-tuning that model. And we used what's called a semantic segmentation approach. So semantic segmentation in computer vision is when you segment the image into different classes. So that's, in this case, we're doing water versus not water. And then you run the algorithm to predict each class label for each individual pixel in the image. So you can begin to see that for each individual pixel in these images, it takes a lot of computational power to do that. And so that's why removing all of the portion of the photo that has nothing to do with the water was really valuable. It saved almost 40% of computational time. So we found the results from this paper. We found that you can see that the prediction of where the horizon is to remove the sky is actually pretty good with some of the models that we were using. However, there's a lot still to do with additional fine-tuning. But what we found is that the model works really well on calm days where it's sunny, not so well on days where it's stormy or the camera is obscured. So that is our next approach. But I want to show you some preliminary video that we have of detecting buoys in the ocean on the next slide. And let's see if I can get this video to work. Here we go. So this is a video of a mussel farm, and the various colors, the boxes, represent the level of confidence in which the model is predicting whether or not an object is a buoy. So the orange and yellow colors are a lower confidence for prediction, but the green colors indicate, I'll play the video again, the green colors indicate a high level of confidence for prediction. So we're moving in the right direction, but you can still see there's some buoys, especially on the far right here, that there's no prediction yet. So we're still continuing to refine our models, but this is a really exciting next step to be able to provide this information to farmers so they can monitor their farms from their computer and decide whether or not today's the day that they need to go out and spend the time and the energy and the fuel to look at their farm, or if they can wait until a better day in the week. Now, I've mentioned earlier that we're also doing work to identify shellfish and harvest assessments. So I wanted to show you a little video of some other work that we're doing with some underwater robotics. So this is let's see, here we go. So this is a remotely operated vehicle. So the larger image in the background is what the robot is seeing. So the smaller inset image is kind of a selfie of the actual robot. But you can see what it does is it goes up and down the water column along those mussel lines and can actually take pictures of each individual mussel if we needed to, and then run that through a model to decide whether or not it's time to harvest that line. And that is really, really valuable information for farmers because normally they have to manually pull up and sample a few mussels from a line, but this way they can actually use streaming video to identify which particular section of their farm they need to harvest this week versus next month. So really, really cutting edge, interesting things happening in aquaculture thanks to data science. So a summary for this particular section here, we can start to use things like sensors and smart technologies to get a better sense of what is happening under the surface of the ocean. And with this data, we can start to integrate and apply advanced machine learning and AI models to start to understand patterns and processes that the data are telling us. But I do want to point out that no matter all of the data that we collect, the real value, the added value comes from the domain knowledge. And that's both from the marine scientists and also the farmers themselves who know the system better than anyone. And so it's really combining the data and the cutting edge analyses with the pre-existing domain knowledge that we have to develop a really exceptional program and hopefully advance the industry in sustainable ways. So I'll leave it there. There's a lot about this project that I have, the program that I have yet to cover. We are currently looking at increasing our graduate student and our postdoc enrollment. So if you're interested in this, if you want to learn more, I've put the website for the program here in green. And I've also put my email below in the footer. You're always welcome to reach out to me with questions there. Okay, so that is aquaculture. I'm going to take you in a completely different direction now. And we're going to go looking at seafood safety in a warming ocean. And this project in itself is part of a larger collaborative project across Japan and New Zealand to essentially map the distribution of harmful algal blooms in an expected ocean changing conditions with global warming. So what I'll show you today is just a little snapshot of some of the work that was presented recently at the 20th International Conference on Harmful Algal Blooms in Hiroshima, Japan. And this is really interesting work that we hope will continue forward both across New Zealand, Japan, and several areas in the tropics that are susceptible to seafood poisoning. So just a little bit of background with seafood poisoning. If you've ever heard of ciguatera poisoning, it's the most common form of non-bacterial seafood poisoning worldwide. This map that I'm showed here is essentially a map of the distribution of the genera Gambierdiscus and the several species that are found within the genera. And it's important because Gambierdiscus itself is the causative agent for ciguatera poisoning, but not all species within Gambierdiscus are known to produce what is called ciguatoxins. In fact, the only known producer in the South Pacific is this Gambierdiscus polynesiensis. And so I've highlighted on this map in red where we actually have known cases of Gambierdiscus polynesiensis and therefore known cases of seafood poisoning. And several of those are in the Kingdom of Tonga and French Polynesia. And then two, the Cook Islands and the Kermadec Islands are actually territories of New Zealand. And so while the species has not been detected in New Zealand yet, its geographic expansion is very likely in a warming ocean. And so there's a real concern that this will be an emerging risk to seafood consumption and the population in New Zealand. And that was one of the primary drivers for doing this study. So when I talk about a warming ocean, I'd like to refer to this figure here. This figure is from the IPCC, the International Panel on Climate Change Global Climate Model Projections. And so this was from their most recent assessment back in 2021. And this figure here shows the expected changes in global mean sea surface temperature between now and the end of the century. And you can see here, this is on the axis, the y-axis is the increase in temperature in degrees Celsius. And these various lines in the graph represent the expected increase in sea surface temperatures based on our carbon dioxide emissions outputs. So what I want to do is just highlight two of these scenarios in particular, this SSP 2.6, which is the shared socioeconomic pathway 2.6. And what that is essentially our best case scenario. That means that if we were to significantly curb our carbon dioxide emissions as of today, we could expect sea level to rise about 1 degree globally by the end of the century. Now I want to contrast that with this SSP 8.6, and that is SSP 8.5, and that is our worst case scenario. And unfortunately, that is the scenario that if we do nothing and we keep everything emissions as they are, this is the trajectory that we can expect by the end of the century. And so that's quite a difference between 1 degree best case scenario and upwards of 4 degrees Celsius warming by the end of the century. And so I think the important thing is while there is some variability between the different scenarios, under all scenarios, we can expect sea surface temperatures to warm, and that in itself is very alarming. So the question that we wanted to ask with this research project, given that we know the oceans will warm, how can we begin to understand changes to the distribution and possible range expansion for this particular harmful algal species, Gambardiscus polynesiensis? And the goal here is to be able to monitor and mitigate that potential threat to seafood safety and human health in New Zealand waters, and then beyond in other areas as well in the Pacific. So there's a lot of work that has gone into this study, and I'm boiling it down to one slide, but I don't think it really does it justice. But for the sake of time, I wanted to just give you a sense of some of the methods that we use to be able to predict these distributions. The first thing that we did was we had to understand what the characteristics were between temperature and salinity that actually promote the growth of the species. And so what we did is we ran laboratory experiments under a variety of temperature and salinity combinations. So these temperatures in this figure here on the right, on the upper side here, show that the various temperatures that we cultured these species in the lab and identified their growth rates to figure out what their maximum growth rate combination of temperature and salinity were. Once we had that, we were able to develop a multivariate regression model from it, and we combined that information with spatial data sets of temperature and salinity for 2 time periods. So we have our current time period, which is for the last 30 years. And what that does is essentially provides a baseline of understanding current distribution for the species. And then we can start to compare that with our future scenarios. And for those, we used our global climate models. And we used those 2 scenarios, this SSP 8.6 versus the SSP 8.5. And just as a refresher of what those 2 were, we have our best case scenario 2.6 and our worst case scenario 8.5. And so when I show the results, those are the 2 scenarios we're looking at. So from that information, the lab experiments plus the spatial data sets, we were able to create predictive models of current and future distribution to begin to assess when and where specifically we need to monitor for future ciguatera poisoning risk within New Zealand waters. Okay, so to give you a sense of the sea surface temperatures that we now typically find off of New Zealand versus what we can expect in the future, I wanted to just show these maps here. So on the left here, we have our historic sea surface temperatures, and I've included a few islands and markers that we've previously talked about because we know that ciguatera poisoning has been found in these locations. And with the sea surface temperature figures here, there's the contour lines. The thick lines represent every 5 degrees change in sea surface temperature, and the thinner lines are every 1 degree change here. So as expected with sea surface temperatures off of New Zealand, we see a warming of waters as we move towards the tropics. And if we start to look and compare that to our best case scenario of temperatures by the end of the century, this 2.6, we can see that it's a fairly conservative amount of warming for our warmest temperatures. So the 25 degree contour line between our current and our future temperatures doesn't really change all that much. We do start to see some changes with our 20 degree contour line. We do see a little bit of a shift forward with warming waters at that point. But if we compare our best case scenario of warming temperatures with our worst case scenario, this 8.5, we really start to see the effects of global warming. So if we look back at that 25 degree contour line, we see that it has shifted actually several degrees latitude to the south. And we're already starting to see temperatures as warm as 30 degrees in the upper portion of our study area. So this is really important because this temperature is one of the driving factors for the growth of this particular species. So what I'll show next is a similar 3-panel figure that shows the distribution, the map distribution for this harmful algal species. So if we look to the left here on the current predictions, based on our laboratory growth rates of temperature and salinity, all of the areas in the red, so the warmer colors, represent areas that our model would predict that we find this harmful algal species. And so for the rest of the figures, the thicker contour line, the white line, represents every 0.1 increase in growth rates, and the thinner line is every half step. So just to show you kind of where those significant sort of increases in growth rates might occur spatially. So as expected, based on what we know from empirical and field studies, we tend to see the greatest distribution of this harmful algal species in the Cook Islands, and secondarily in French Polynesia and the Kermadec Islands. But at this current time, we don't see any growth that occurs near the New Zealand coastline. But what happens is if we use those predictions with our global climate models and look at our best case scenario into the future, we don't really see much change in the maximum growth rate band. It still hovers around the Cook Island, but we're starting to see, you can see at the very top of the North Island, we are starting to see conditions that are hospitable for growth for this species. And that is very concerning, especially if we look on the far right panel to our worst case scenario. And that is the scenario that we expect if nothing changes with our CO2 emissions. This is the scenario that will most likely occur. And in this scenario, we do see growth rates that occur throughout most of the North Island and New Zealand. And while they're still fairly low growth rates, what this means is that this species can actually survive in these waters, and our likelihood of finding the species is greater in a changing ocean. So based on these two predictions, the SSP 2.6 and the SSP 8.5, I was curious to see what sort of the spatial difference is if we compare these two predictions, these scenarios in the future. So this next figure here, the first two panels on the left here are the same figures from the previous slide. Those are the predictions moving into the future for this harmful algal species. But what I wanted to do is show the sensitivity analysis, or the spatial difference between our best case scenario and our worst case scenario. And what this really shows is that the warmer orange and red colors here really indicate the area, the spatial extent where we're going to see the greatest change in growth rates between a best case climate model and the anticipated as is worst case scenario. And so what this tells us, this sort of band that extends longitudinally across the North Island, is that this is where we can expect to see the presence of this harmful algal species in a warming ocean. And what's concerning is that for New Zealand, the majority of the population lives on the North Island. And so the risk of seafood poisoning for the population is the greatest in this area. So that is a very quick overview of some of the work that we're doing with predicting harmful algal species distribution in a changing ocean. I haven't even begun to share the results of our Japan model as well, but hopefully a paper will follow soon to share. But to summarize the key findings from this work is that under all scenarios of global climate warming, whether it's the best case scenario or the worst case scenario, it's expected that the future warming will affect the distribution of this ciguatoxin producer, Gambierdiscus polynesiensis, causing a change in distribution and range expansion. Growth conditions will likely shift poleward and increase the risk of seafood poisoning in New Zealand waters. And so what we can do is start to use these predictive models to develop more accurate risk assessment tools so that we can begin to anticipate and mitigate this threat. What we would really like to do is move into seasonal and near real-time predictions so that we can accurately tell folks what times of years and under what conditions they may want to avoid eating certain types of seafood to avoid contamination. So that's a summary of the work that we are doing here in New Zealand. And I just want to say a big thank you to all of the collaborators and colleagues and the organizations that have funded this particular project. And please stay tuned for more from this if you are interested in harmful algal blooms. But what I want to move to next is the final segment of this presentation, and that is a legacy project that I've been involved with for many years and am excited to share the next chapter of this work, and that's looking at dynamic ocean corridors for North Pacific loggerhead sea turtles. So what I would like to do first is just walk you through some of the previous research that we have published that tries to understand the individual movements of animals from this population, and then finish up the talk with the next steps that we're doing to actually run the first sort of experimental oceanography approach of its kind ever known for any migratory species. So let's first start with a little bit of background for North Pacific loggerheads. So for a long time, loggerheads were known to be found on both sides of the North Pacific Ocean. So these green tracks here indicate historic observations of loggerheads, both in Japan as well as the Baja California Peninsula in Mexico. But for a long time, we thought, well, these have to be two separate and completely distinct populations. If you know much about sea turtles, understanding their life history when they're very young is very difficult, because turtles leave their nesting beaches as little hatchlings and they disappear into the ocean. And then they emerge years or decades later somewhere else, completely different. And we have, it's a mystery as to where they've been all that time. So I think between not having a lot of information about their early years, and at the time thinking, how could one individual actually swim across an entire ocean basin, it was assumed that these must be two completely separate populations. But through a combination of satellite tagging and genetic analyses, we now know that individuals from this population represent one complete and unique population of North Pacific loggerheads, which is just truly phenomenal to be able to connect all of the animals on both sides of the ocean basin. So we know that we now know from a lot of the studies that have been done that individuals from this population leave their nesting beaches of Japan. So Japan is the only location in the entire North Pacific that loggerhead sea turtles nest. And so all loggerhead sea turtles that we know of come from nesting beaches in Japan. The hatchlings leave those beaches and then spend their juvenile years moving across the North Pacific Ocean through transition zones of productive water. Now, because sea turtles are a long-lived species, their juvenile period, the time at which they are moving through the open ocean, it can last up to 20 or 30 years. Pretty amazing for an animal. And once they're ready to mate, all individuals from this population will return to the Western Pacific, nest on Japanese beaches, and then we believe adults stay within the Western Pacific off of Japan and the South China Sea. So we know that a subset of this portion of the population as juveniles spend a lot of time in the central North Pacific Ocean. You can see a lot of the tracks in this figure represent a lot of time spent from a lot of turtles in the middle of the ocean. But we do know that some individuals will transition over to the Baja California coast. And in fact, we know that Baja California is a hotspot for juvenile loggerhead sea turtles from aerial surveys that have estimated tens of thousands of individuals that are year-round residents of the areas of Bahia Ulloa year-round. And again, because juveniles can live, the juvenile stage can be 20 or 30 years, sea turtles can live off of the coastal waters in this habitat for decades until they're mature enough to return to their Japanese nesting beaches and again remain in the western North Pacific. So no matter where they go, whether it's the central North Pacific or the coastal waters of Baja California, this journey, this transoceanic journey represents a very unique, once in a lifetime event for individuals from this population. So it's a really important migration that only occurs during their juvenile stages. However, throughout all stages and locations of their oceanic range, loggerhead sea turtles, like most sea turtles, are subject to high rates of fisheries-related mortality. So while we have knowledge of their movements within their core habitats, both from the oceanic habitat and their coastal habitats off of Baja California, it's important to know that our understanding of the connectivity between these completely separate and different habitats, these locations, is still very fragmented. And for this reason, because we don't have a complete picture of their migratory movements and patterns, that leaves us with critical gaps in our ability to effectively assess and protect this mysterious and endangered species. What's even more concerning is that in recent years, loggerheads, very small loggerheads actually, have been found in large numbers off of the United States West Coast, so within the Southern California Bight here. These occurrences, these purple tags, occurred during exceptionally warm conditions from the 2014-2015 El Nino, and then again with the marine heat wave in 2015 and 16. And this has been a real concern because there are fisheries that operate out of this particular Southern California Bight area that, given interactions and overlap with juvenile sea turtles, it has been of conservation concern. So given all this information, the question that we wanted to ask were, what are the mechanisms that allow a turtle to go recruit from the open ocean in the middle of the North Pacific all the way to its coastal habitat off of Baja California? And while we have enough tracks to start to get to paint the picture of how they make this transition, we still don't know the mechanisms for this coastal recruitment. And this next slide, I think, really sums up sort of the question. And the slide is from the 2021 Frontiers in Marine Science paper that we published, but the wonderful additions have been added by Dr. Alberto Abreu, who is joining us today. And so I really like this figure because I think it really encapsulates the questions that we sought to answer with our research. As we know that individuals will spend several years, maybe more than a decade, in the foraging oceanographically in the central North Pacific, but only a portion actually continue eastward and transition over to neritic or coastal habitat off of Baja California. We have over 231 satellite tags that have provided information from the 1990s and early 2000s, but we still have so many questions of how and what are the factors? Are they currents? Are they magnetic fields, temperatures? We know temperature is for sea turtles who are driven by sea surface temperature. Temperature is a really important factor. So we thought to explore the temperature component a little bit more. And what I'd like to show you with this figure is that really with the 231 satellite tags that we had, only 30% of individuals actually migrated over to the Eastern Pacific Ocean. And given that only 30% migrated into the Eastern Pacific, only 3% or 6 unique turtles actually continued further and moved into this area of the California Current, which is the shaded polygon off of the North American coast. And I've highlighted these important 6 tags, color-coded them. And what this really tells us is that we have a large data set of North Pacific loggerheads. We know that tens of thousands of loggerheads are found off of the Baja California Peninsula, but only 6 have actually shown us what it is, where these movements, how turtles actually transition over across from oceanic to neritic habitat. And so given that we know temperature is an important driver, what we did was we explored the ocean conditions that corresponded to the movements of these color-coded turtles here. And we found that individuals who traveled the furthest to coastal habitat did so in the warmest sea surface temperatures. So we began to think that temperature and ocean conditions played a very important role in a possible recruitment to coastal habitat. But we wanted to provide additional information because a sample size of 6 is still very small. And so we sought out an independent data set using stable isotopes and skeletochronology from the bones of stranded sea turtles along the Baja California Peninsula. And they just so happened, these bones happened to overlap with the satellite tagging data set time period that we had. So that helped us get a greater sample size and be able to understand the transition from a stable isotope perspective of carbon to nitrogen, of the years at which turtles transitioned over from oceanic to coastal habitat. And so between the satellite tags and the stable isotope analyses, we were able to build a larger sample size of relative recruitment per year. And what we did is we matched that with the ocean conditions for those years, and we developed a hypothesis called the thermal corridor hypothesis, which basically states that under certain ocean conditions, exceptionally warm ocean conditions, we see intermittent migration of sea turtles, recruitment to the Baja California coast. And so in the next figure, I want to show you a figure that came from the paper, and I think this does a good job of really showing the difference in ocean conditions. And so for this figure here, for panels B and D, we have warm years. So whether that is a marine heat wave or an El Nino year, both in the winter preceding recruitment from our satellite data and stable isotope information, and then the summer of that particular warm year. And you can see that ocean conditions are much, much warmer in that area in the box of which we know turtles transited through, especially in comparison to cooler years, for example, La Nina years. And what this really told us is that exceptionally warm conditions may provide those cues for turtle in the central North Pacific to access an intermittent corridor that opens up, this warm corridor that opens up under warm conditions. Those conditions would be anything, a 3-month average of sea surface temperature anomalies above 0.5 degrees Celsius, which corresponds to how El Ninos are essentially forecasted for a given year. And what this really does is this thermal corridor would allow for an increased number of sea turtles to make their way to recruit to coastal habitat. And what we think is that under this hypothesis, the thermal corridor opens up and more turtles can access their developmental grounds off of Baja California. And in years when it's too cold or there's not enough warm water, then turtles will turn around and stay within the central North Pacific and maybe wait for another year or 2 or 5, because their juvenile life history stage is so long, they have enough time to basically wait out another warm year in the future. So with this hypothesis, we published this paper and we thought, well, you know, we had 231 satellite tracks over 15 years. When would we ever be fortunate enough to have a data set like that again? How can we actually go to a funder and say, please give us money to tag more turtles over the next 4 or 5 years under ocean conditions? But I'm very pleased and proud to say that we actually get to test our hypothesis. So the next phase of this project is called the Sea Turtle Research Experiment on the Thermal Corridor Hypothesis. And what we call that is Project Stretch. And this is really a novel experimental oceanographic study that will allow us to test the thermal corridor hypothesis by releasing 25 turtles over the next 4 years in the North Pacific Ocean, which is 100 turtles altogether. It just, even saying it, sometimes I can't believe that we actually get to do this. So we'll satellite track these turtles, and then what we hope these turtles each year will experience slightly variable ocean conditions, anything from warm years to cool years and neutral years, so that we can very thoroughly validate and test this hypothesis. And what we hope is that by understanding when and where this corridor opens for this species, we can start to develop dynamic management approaches for species conservations for sea turtles and for all of the animals that might actually use this corridor as well. Okay, so I wanted to show you some of the figures of the project thus far. So in order to have 25 turtles a year to deploy into the central North Pacific, this project would not have been done without the exceptional care and the rearing of hatchlings from the Port of Nagoya Aquarium in Japan. So the picture on the left shows a baby sea turtle hatchling that was raised from birth to 2 years of age. So each turtle that we release every year will be about 2 years old. So the process begins in advance. The turtles that were released this summer were raised in 2021, and the aquarium is currently raising next year's turtles as well. So it's an invaluable part of this project. So before we released the turtles, we had to go over and prepare them for their big journey ahead. So in June of this year, we went over to the Port of Nagoya Aquarium in Japan, and we assisted the aquarium with outfitting each of this year's 25 sea turtles with satellite tags. So each turtle was outfitted with a resin fiberglass method tag, which involved a lot of care and cleaning of the turtle shells, and then the application of the tags. And the red circles on the tags are, it's a biofouling agent, so it prevents any sort of fouling or organisms that would attach around the base of the tag. It was a really a fun experience and really special to see these turtles before they were released and made their big journey. So the question leading up to the release was, well, where and when do we actually release these turtles in the ocean? We had to balance a couple of factors. The primary factor was the welfare of the animals. We had to make sure that sea surface temperatures were warm enough for the turtles to be released at a given time of the year. We had to balance that with the experimental design approach. So we wanted to release them in a location that was representative of where they would naturally be that time of year. Because if we released them somewhere completely different where they wouldn't be found, then it wouldn't really be a good way to test the hypothesis. We also had to balance that with finding someone to actually transport the turtles into the middle of the ocean and release them on our behalf. So a lot of logistics went into this, and the location in red here was the final location that the turtles were deployed. And a lot of thought went into how we can do this to balance the temperatures, the battery life of the tag. We didn't want to release them off of Japan because it could take several years to decades for turtles to actually make their way across the ocean. And at that point, we may have lost the battery life of the tag. So this was our final destination, and this box here is our thermal corridor, which we will hopefully see in the near future the turtles moving through. So in order to get the turtles into the middle of the ocean, we partnered with a ship of opportunity. In this case, that was a cargo ship called the Galaxy Ace that just so happened to be moving through the North Pacific. And this was a valuable partnership because they made this open ocean deployment happen for us. So what I'd like to show you is a very short video of one of the turtles that was released here, and I'll just play it in the frame here. So you can see the turtles, each turtle was dropped down in a basket from the side of the ship and then gently released by tipping the basket over. And what you can see is this turtle very happily begins its ocean journey into the North Pacific, swimming away. And so one down, 24 more to go for them. Each turtle, before they were released, were given a name. And this was really special because it allowed us to select names that came from the various regions by which a sea turtle, a North Pacific loggerhead, transitions through, from Japan through Hawaii, Southern California, and Baja California. I'm proud to say that the names chosen to honor the 5 truly wonderful conservation heroes who've worked really hard in Mexico for sea turtle conservation: Antonio, Betty, Victor, Maricel, and Chuy. We have been following their turtle tracks, and if you're interested in following along as well, on our website here, loggerheadstretch.org, you can actually follow the individual tracks for each turtle. It's a map that's updated on a daily basis, so you can look online every day to see how far your turtles have moved. If you have a special favorite turtle that you want to pick out and track them, you can actually follow along and select their name, and then you get a filtered version of that individual track. So a really neat map to look at. So now that the turtles are tracking, we're interested in what are the environmental conditions that they're experiencing. So from their point of release, this black X here, we've been tracking their daily movements in relation to sea surface temperatures. And you can see that this white line represents the 18-degree sea surface temperature isotherm, and that's an indicator of productivity for the ocean. It's a convergent zone where you have the waters from the subtropical gyre that meet the subarctic gyre, and they produce a lot of productivity and convergence of prey species. So the turtles left their cargo ship and all headed north as expected, because this productivity isotherm basically shifts north seasonally. And then by September, we can see that isotherm begins to shift south again. So our turtles have moved north, they've moved south, they're now moving south, and the big question is where will they go next? And we've been keeping an eye on several turtles in particular who have shown a particular inclination to move eastward, these turtles here, and we'll follow along. What's really interesting is that we had hoped that under the 4-year turtle release program that we have, that we would get an El Nino or a La Nina, and right out of the gates, the very first year, we are now experiencing an exceptionally warm El Nino coupled with a marine heat wave in the North Pacific Ocean. So this is our first year of release. It is our proof of concept year, and it's turned out to be quite an exceptional year to validate this hypothesis. And so what we hope next is that by the year 2027, we have a La Nina year to really compare the movements of each year's release. So I'd like to just end this talk by promoting some of the great work that's being done for this particular research project. We have a website called Loggerhead Stretch, found at loggerheadstretch.org, or you can use your phone to scan the QR code here, and it'll take you there. And there's a lot of information that I wasn't even able to cover in this talk today. You can learn more about the background of the project. You can meet the team and see all the various colleagues and contributors. And you can follow along with a map. We have the website is in English, Spanish, and Japanese. So it's already prepared and ready to go for your viewing. And what we're currently working on is an education and outreach component. And that's a really exciting section that we're looking forward to developing more. It has resources to learn more about sea turtles. It has infographics for how population dynamics work and migratory movements for this population. And then really interesting sort of individual cards of each turtle that was released and some fun facts. So we also have a social media component, so you can follow along on Facebook, Instagram, and X, formerly known as Twitter. So a lot of good stuff. And probably the thing that I'm really enjoying the most is the blog that has been set up by one of our team members, Mark Rice, who has done a fantastic job of capturing what's happening with the turtle tracks and the oceanography, and really providing up-to-date information. So if you're curious about what's happening and the science behind it, I would really encourage you to check out the blog that we have with that. So a final note, we are continuing to follow our turtles, and I'll leave you with a question of who will make it to Baja first and when will they get there. So stay tuned for more. And I think in the next few months, by the beginning of next year, we'll see some really exciting developments with these turtles. So in summary, what we can do is begin to look at changing ocean conditions and find ways to dynamically manage them to protect the species. Hypothesis validation is an important step, and Project Stretch, we hope, will really expand our knowledge scientifically, educationally, and for conservation. So I know I'm running short on time, so I will just show a picture of all of my wonderful team members and colleagues, and leave it there, I think, with a summary. So thank you very much.
Thank you so much, Dr. Briscoe, for this amazing and very complete presentation. We had a closer look at the work you developed in New Zealand and these details about Stretch project. Thank you so much. So let's move on to the questions and answer session. Empecemos con esta sesión de preguntas y respuestas. Recuerden que lo pueden hacer por el chat, pueden levantar la mano, de preferencia, si se puede, que lo pudieran hacer en inglés. Si no, tenemos al doctor Abreu, que también nos puede apoyar para la traducción. Y veamos en el chat, Nacho, ¿nos ayuda si hay algo o hay alguna mano que yo no alcance a ver?
No, en el chat no hay nada, ni manos levantadas.
Ok, mientras tanto, no sé, doctor Abreu, si usted quisiera comentar algo, porque bueno, también el doctor Abreu y la maestra Raquel Briceño están participando en este proyecto. Entonces, no sé si usted quisiera agregar algo, doctor, en lo que llegan las preguntas o los comentarios.
Pues a mí me da mucho gusto tener aquí a Dana con nosotros. Es un proyecto en donde estamos participando, pues muchos colegas de Japón, de Hawái, de California, y la verdad lo curioso es de que no nos hemos reunido en persona, para nada. Entonces, los que más se han reunido, los suertudos que les ha tocado ir a Hawái, por ejemplo, para estar con las tortugas, los colegas japoneses que han trabajado con las tortugas desde su nacimiento. Pero pues vean lo que se ha logrado y es un proyecto pues de muchos recursos que está abordando preguntas bien interesantes que a todos nos interesa. Y hay que decirlo, y aquí creo que también hubo una participación del grupo tortuguero, que en México ha trabajado muchísimo en pro de la conservación y la investigación de esta misma especie en su hábitat en México, en donde, como lo decía Dana, hay una concentración de grandes cantidades de la fase intermedia del desarrollo, ¿no? Los juveniles y preadultos. Y es de gran preocupación para México porque ahí también hay una alta concentración de pesquerías y desgraciadamente también una interacción pesca incidental y mortalidad por lo mismo, ¿no? Entonces, si bien el proyecto que nos presentó Dana habla acerca de qué es lo que pasa para que lleguen a México, a nosotros también nos interesa saber qué pasa después, ¿no? Entonces, como que hay un colofón de qué es lo que sucede con las tortugas después que nos interesa mucho. Y el grupo tortuguero que está activamente trabajando con el monitoreo de las tortugas en áreas de alimentación, ¿no? Dándole seguimiento, conociendo su distribución, tasas de crecimiento, las tasas de mortalidad a través de los varamientos. Todo eso es parte de un rompecabezas muy grande y aquí entonces cada quien pone su granito de arena, contribuye con una pieza del rompecabezas para tener una mejor comprensión y lograr entonces tener con esa mejor comprensión, saber cómo orientar las actividades del manejo, de la conservación, de las actividades pesqueras, como lo decía Dana, poder integrar ese incremento en las capacidades técnicas para poder hacer una mejor predicción, ¿no? Y una mejor conservación y manejo, ¿no? Entonces, pues estamos muy emocionados, todos muy a la expectativa, como se vio en esa gráfica de Dana, ¿no? O sea, por allá están y se ve fácil a distancia una vista desde el cielo dónde están, pero todavía les falta 1800 kilómetros por recorrer, y pues es una volado a ver cuántas de esas vienen para acá y cuánto tiempo van a tardar y si les alcanza la batería.
También, claro. Ok, muchas gracias. Gracias, doctor. ¿Alguna pregunta, comentario? Would you like to add something, Dr. Briscoe?
Yeah, I want to say thank you to Alberto. That was a really great summary. I caught most of it, but I really appreciate that the detailed explanation with that. And yeah, we're very excited about this work, and there's still so many questions to answer for juveniles of this population. And just really interested to see if they do go to North America, where they will wind up. And what we're finding is that turtles from this year have traveled further north than any of the previous tracks. And what does that mean for how, when they start to move down to the North American coast, how does that impact their habitat distribution from there? And so we're already seeing the possible effects of climate change based on information from this year. So, yeah.
Thank you so much, doctor. Muchas gracias, doctor Abreu. Si ya no
No,
hay más...
yo tengo una pregunta.
Sí,
I have a
adelante.
question. All right, Dana, if you were to bet, what proportion of turtles of the 25 would you bet on arriving, reaching Mexican waters?
I would, my dearest hope is all 100%. Yeah, I would like that. I think what we're seeing with the tracks is that there's a sort of a portion that have very clearly indicated that they're headed directly to the North American coast and to Mexico. We're seeing some that have kind of taken a turn and are moving into the central North Pacific. And that'll be really interesting to see if those tags, I mean, what we really hope is from the historic data set, we got almost 5 years out of an individual tag. So we would ideally hope that we get at least 1 complete year of tagging information, but maybe more. And so it could be that some of the turtles who may not make it this year to Baja, they may wait until next year, or they may need to get a little larger and make their way over. So my hope is that it may not be this year that they all directly go to Baja California, but that all 25 find their way at some point in their tagging lifespan.
Thank you.
Okay. Gracias, doctor. ¿Algún último comentario, pregunta? Creo que
I
no.
do see a question. I think this might have been a direct message to me. There was a question about how many images were used in the mussel project to do the computer vision analysis, and if there was a special program to work with the image or video. So I'm happy to speak to that. The images that we collected, so we collected videos, and what we do is we analyze frame by frame the video. So you can imagine that if we have a week's worth of video on the farm, the sheer number of frames that we have from a week's worth of videos is just massive. And so that's really where using these off-the-shelf sort of these pre-trained models can be really valuable, because they allow us to shortcut the step of having to have someone label large amounts of frames within these videos. And so we're able to expedite the process of the object detection by because we have probably I don't know how many gigabytes of data and images we have, but you can imagine having to spend your days looking at each individual frame. It would get overwhelming and boring pretty quickly. So yeah. And the program that we used was Python for that, if that's helpful to know.
Okay. Thank you, Dr. Briscoe. Doctora Tomé, sí, adelante.
Le abierto el micrófono, pero
Audio,
ay,
no le escuchamos, doctora. Ahí está, otra vez. Otra vez, doctora, dele por favor clic en el microfonito. ¿No lo permite? Ahí está.
Ya,
ya, creo que ya.
Ya, adelante, doctora.
Okay. Hello, Dana. This was congratulations. It's fantastic work. And to me, I entered a bit late to the talk, but you have all these different projects, and I was wondering, what would you say is the linking thread that makes, I don't know, for example, if it's the diverse means of acquiring biological data, I mean, data of biological value with different resources, no, like satellite images, so that this links all the projects that you've been working on.
Yeah, that's a great question. I would say that the overarching link is our ability to acquire different types of data sets and really begin to synthesize them in ways that we can start to understand the effects of climate change with them. I think now more than ever, we have a vast amount of data at our fingertips. Even in the way that we use our smartphones, we are just constantly, generating data. And so what we hope to do is to bring those technologies into the marine science sphere so that we can actually have the data drive those decision-making components under changing ocean. Yeah.
Tu micrófono, doctora.
Sorry for the technological delay. Yeah, it's amazing, no? How, I mean, to take advantage of all the data that already exists and to put it to work to answer questions about climate change. So that's amazing. Congratulations. Thank you.
Gracias, doctora Tomé. ¿Vemos
aquí.
Sí, dime, Nacho.
Ingrid tiene dos preguntas, pero las mandó al chat en español. Dice que si hay alguna distinción de sexos en las tortugas liberadas, y la segunda, que si hay, espera algún comportamiento dependiendo del sexo del individuo.
¿Nos ayuda, doctora Abreu?
Sí, claro. Did you hear those questions, Dana? The first question is whether, is it anything known about the sex of the turtles that was released? And if there is any expectation of different behaviors by the different sexes? Oh,
that's a great question. I do know, so George Balas is one of our co-investigators on this project, and he has a lot of experience in sea turtle biology and ecology, and he would probably be the person to really formally answer that question. I do know in recent conversations that we've had with him is that based on the small size of the turtles upon release, we don't know yet the sex of the turtles, and it might, given that, it would be really interesting to find out if we did know that, how that would differentiate. I think the short answer is that we don't have the information on the sex, and so we can't use any of that to extrapolate what behavioral differences might be at play here. But I think that would be a really interesting thing that we could hopefully do in the future. If I don't know if maybe in tracking these turtles, if anyone happens to come across them in the ocean in a few years, if we can get that information. But I think at this point, we don't know that. So we have to assume that we're working with that limited information on that. I don't know, Alberto, if this is a good time to raise the question too. One of the things that we've discussed is whether genetics are at play with turtles, whether they make that decision to stay in the Central North Pacific, or if turtles that nest from a certain beach in Japan may be more genetically inclined to be the ones that make it to Baja California and the North American coast. And that's something that we really hope to investigate as part of the larger goals of this project as well.
Sí, yo quisiera agregar algo. I will speak slowly, see if you can follow me, Dana, see if you agree. El sexo de las tortugas chiquitas no se puede determinar visualmente. Se puede hacer alguna inferencia a través de análisis hormonales y otros factores. No se hizo, pero se tienen muestras de sangre de todas las tortugas. Entonces, si bien es complicado el usar esa muestra de sangre para ese análisis, es algo que a lo mejor, Dana, podríamos hacer. Entonces, el enfocar la pregunta de si hay alguna diferencia de los comportamientos entre sexos, pues se pudiera abordar. Y el otro factor interesante es de que las tortugas marinas tienen una tendencia en algunas especies, algunas poblaciones, a que haya más de un padre en cada una de las camadas, de las nidadas. Es decir, este grupo de crías tuvieron una madre, pero puede que tengan más de un padre. Entonces la contribución del padre pudiera ser de los padres, que entonces también agrega otra dimensión interesante que igual por genética pudiéramos incorporar. Great, more work from me.
That was a much better answer, Alberto, than the one I gave, so thank you.
Gracias. Thank you, Dr. Briscoe. Yo creo que cerramos con esta pregunta, si bien no hay alguna otra. Creo que ya no. Recuerden, les invitamos a seguir el proyecto Stretch, que justo la doctora Briscoe nos enseñaba pues todo lo que se está haciendo, el blog, toda esta cuestión para que pues no nada más se quede en el ámbito científico, sino que también todo el resto del público también lo pueda ver y ser partícipe. Thank you so much, Dr. Briscoe. We're really happy to have you here. And okay, thank you, Doctor. Here it is. Ahí está la liga para que la puedan seguir. Y pues muchísimas gracias. Este es el penúltimo seminario de nuestro ciclo. Ya tendremos la última sesión que justo toca para presentar a la nueva académica que está en Puerto Morelos. Pero en cuanto a este eje temático, pues bueno, con estas dos sesiones que tuvimos del doctor Abreu y la doctora Dana Briscoe, pues bueno, cerramos este eje temático. Recuerden que si se lo perdieron o entraron un poco tarde, lo pueden encontrar en nuestro canal de YouTube, que lo subiremos próximamente. Muchas gracias, doctora Abreu. Thank you so much, Dr. Briscoe.
Muchas gracias. Thank you so much for this opportunity today.